ENIGMA-ADHD studies brain differences related to ADHD across the entire lifespan. And currently we consist of 34 cohorts, and this represents 4,000 subjects, healthy controls and ADHD patients.
What you see in ADHD is that most of the studies are really only about children. And as in many other fields of neuroimaging, the samples that have been analyzed are very, very small. So that's really a problem. There have been a few meta-analyses, but these suffer from the problems that everybody uses a different method to segment, etc. So what we were hoping for with doing this in ENIGMA was really to bring the field together, have the field work together, look at lifespan data, look at larger samples and combine something with more consistent methodology.
In ENIGMA-ADHD, we analyze brain differences in children separate from adults with ADHD. And in children, we see the most pronounced effects, so the largest differences between the brains of children patients with ADHD compared to healthy controls. And in the adult group, those differences seem to disappear. So what we show is actually a delay of maturation in the subcortical regions of the brain. But they are normalized actually in adults with ADHD. So there we do not find the differences anymore.
Amygdala is also affected and actually has the largest effect size in ADHD. So the effect sizes that we see in ADHD, but also in autism, for example, are relatively smaller than what you would find in schizophrenia. But the amygdala is actually the largest effect size, surprisingly. But I guess because the samples until now have been rather small, that hasn't been found.
We use methods, both meta-analysis as well as mega-analysis. So we collect individual brain data from patients and controls, where we can perform meta-analysis, so we keep the structure of a study in function, or we can pull all the individual data, and this gives us the opportunity to also study effects of age and clinical effects on the brain.
What is particularly important also in ADHD is the cross-disorder analysis, especially where we're talking about these childhood onset analyses. So the cross-disorder analyses can really tell us whether there is overlap or differences between different disorders. So we can, because we have used the same methods, we can easily put the data together and see where data overlap and where there are really differences between the different disorders, and how that could help us, for example, to differentiate certain disorders at an early time point. There is great heterogeneity in ADHD, and that is one of the things that we're now also addressing by, for example, trying to find subgroups within the data and see whether these are somehow also at the phenotype level distinct from each other.
In ENIGMA, many disorders of the brain are studied. Those are psychiatric disorders such as schizophrenia, bipolar disorder, depression and also ADHD, and neurological disorders such as epilepsy.
Everybody can join. We're on the website and the contact is on the website. You don't need to give us your data. That's the good news. We do share data within the group, but it's not mandatory, so it's very easy.